Hello. So for my protein project, my assigned protein is acetylcholinesterase, and the mutation, which is not really a mutation, it's more of a chemical change, is when sarin, also known as snake venom, binds to the protein's active site. So acetylcholinesterase is an enzyme that facilitates the hydrolysis of acetylcholine into choline and acetic acid. I categorize this as an enzyme because its main function is catalytic activity, which is just breaking down the acetylcholine. So acetylcholinesterase is found in the synaptic cleft of nuclear muscular junctions, and the synaptic cleft is a small space where neurons communicate with each other. And so that is found in neurons and muscle cells, and then the neurons and muscle cells are found in animals, including humans. So I categorize the type of change the mutation has on the protein function as the protein gaining a new function, because it stops when the sarin connects to or binds to the acetylcholinesterase or to the active site of acetylcholinesterase, it stops the normal enzymatic function by permanently blocking the active site. So the main functional group present in the original AA side chain or amino acid side chain is a hydroxyl group, which is found in the amino acid serine. Serine, which is found in acetylcholinesterase's active site. The serine side chain is hydrophilic because the hydroxyl group inside of it is polar, which allows it to form hydrogen bonds with water and other polar molecules. And the side chain is also neutrally charged. I would categorize the serine side chain as small compared to other amino acids because its size allows it to fit into the active site and interact with the substrate. So serine's hydroxyl group allows it to form hydrogen bonds with nearby amino acids, and it allows it to act as a nucleophile. And a nucleophile is a reactant that forms covalently by donating a pair of electrons. So for the mutated side chain, the main functional group found in the mutated side chain is the hydroxyl group found in serine is phosphorylated by the sarin. So the main functional group found in the mutated side chain is a phosphoryl group. So that binds with the hydroxyl group that is already attached to the serine, not the sarin, the serine, which is the amino acid. And so that just turns it into a phosphorylated serine. So the phosphorylated serine becomes hydrophilic, and it becomes very hydrophilic, and that prevents it from functioning as a nucleophile. The mutated amino acid side chain has a negative charge because of the added phosphoryl group, and the phosphorylated serine is large, which blocks the access to the active site in the acetylcholinesterase. So the normal serine takes part in hydrogen bonding and is able to perform catalytic activity through its nucleophilic hydroxyl group, while the phosphorylated serine can't act as a nucleophile because of the covalent bond that it created with the sarin, which is the snake venom. So it also disrupts acetylcholinesterase's ability to interact with acetylcholine. So the chemical change affects the protein function because the phosphorylation of the serine by the sarin inactivates the acetylcholinesterase. So this makes it so that this acetylcholinesterase cannot hydrolysize acetylcholine, causing the acetylcholine to accumulate in the synaptic cleft. And this affects the cell, causing the acetylcholine to build up in the cell, overstimulating nerves, and causing muscles to constantly stay contracted and overactive. And so this can affect organisms by causing symptoms such as muscle spasms, respiratory failure, and potential death if untreated because of the buildup in acetylcholine. So now, here's my 3D model. So I tried to depict the alpha helix structure of the protein with the ribbon around my green circle, and so for both. And so these are pretty much the same, or they have the same structure. And these are both the acetylcholinesterase before it actually before it hydrolyzes the acetylcholine, so turning it into the acetic acid and the choline. So this is just like how it is normally, I guess. So right here, the yellow circle or orange circle is the active site, which contains the serine. Yeah, the serine. And then this pipe cleaner the pipe cleaners for both are the other amino acids. And so the acetylcholine would come into here, and then it would be broken up using the serine, and then with the help of the other amino acids that are like histidine or histidine, how you pronounce it, and then guanine. I think that's how you pronounce it. So for the mutated version, instead of the acetylcholine coming in and then having it comes to the active site and then the active site hydrolyze it, the sarin, so the snake venom, it comes in and it blocks it, like it changes the shape, so it blocks the active site, like so you can see the connector right there. It blocks the active site, so if the acetylcholine did come in, it wouldn't be able to be hydrolyzed. It would just be floating around. So yeah, there's my project. So 3 limitations to my model is that 1, it's more simplified, So it doesn't actually show what happens when the acetylcholine actually does come in, and it doesn't show the how the acetylcholinesterase will actually break that down versus, or yeah, break that down versus how it wouldn't break it down, or how the mutated version wouldn't break it down. Another limitation is that I wasn't able to show like the true structure. I guess I did try to um, show it a little bit with the alpha helix structure in the with the ribbon, but when the actual structure, it's just like a bunch of alpha helix structures and then like a couple of like the beta structures. And this is just the alpha helix. I just show here the alpha helix structures, and then I have kind of have like the clay ball right there, or circle, to kind of actually show the active sites and then the amino acids in the sarin. Another, yes, another limitation to my project is that it also doesn't show how it affects the body, or so if, like you can see how it may affect this, but it doesn't show like um, the broader effects of the mutation or the chemical change in the actual cell. So yeah, those are my limitations.